Hello everyone, welcome to today's vampire seminar. We have Maliha Ahmed here with us today. She's recently completed her PhD with Dr. Wang from the University of Waterloo, where she mainly focused on models of thalamocortical systems. She's now a postdoctoral associate at the Picower Institute for Learning and Memory at MIT, where she works with Dr. Henry Brown. And today she's going to talk to us about multiscale modeling of neurosteroid-mediated seizure trajectories in childhood absence epilepsy. Take it away.
Thank you so much. Thank you so much for being here, everyone, and thank you, Dr. Wang, for the kind invitation. This is my first invited talk since moving here, so I'm pretty excited about the opportunity. I think Sreshta pretty much covered everything that I would have said about myself, so I guess I don't need to take time to introduce myself in that regard. But yeah, so this is my part of my doctoral research that was recently completed earlier this year. So Professor Sue-Ann Campbell, I've worked with her on thalamocortical network models since my time as a master's student, and then continued some of that work and extended it beyond what we did during the master's into my doctoral research. So the title of my talk is Multiscale Modeling of Neurosteroid-mediated Seizure Trajectories in Childhood Absence Epilepsy. So I think it makes sense to start with discussing what CAE is. It's an idiopathic generalized epilepsy syndrome, affects children between the ages of 4 to 12 years. Interestingly, it's more prevalent in girls. Its genetic etiology is linked to mutations in ion channels as well as neurotransmitter receptors. It is characterized by these brief periods of impaired consciousness, which can occur multiple times a day. And often they're mistaken for a child just daydreaming because they will tend to just stare blankly into space. These seizures are non-convulsive, so there's no motor activity involved as such. And many a times, actually, teachers might be one of the first people to notice it in a child and then inform the parents, which can eventually lead to a diagnosis. Clinically, they're diagnosed using an electroencephalogram, which are these scalp electrodes that record brain activity and can give an understanding of underlying brain activity, like brain rhythms and oscillations. What the EEG shows in the context of absent seizures are these bilateral synchronous 2.5 to 5 hertz spike wave discharges. And this pattern is very characteristic of an absent seizure. The most confounding feature of CAE is its ability to resolve in adolescence in most cases. And given that remission often occurs during adolescence, which is also a time of onset of puberty, the association of sex steroid hormones is also suspected. And this association is actually further supported by this phenomenon called catamenial epilepsy, which is seizure exacerbation at particular times during the menstrual cycle. Now, that is not specific to just CAE or just absent seizures. This is something that's noted in women with other epilepsy types as well. And in fact, progesterone and its metabolite, allopregnanolone, have been shown to exacerbate absent seizures in both non-remitting human models as well as rodent models of CAE or of absence epilepsy. In animal models, what we see is an increased SWD incidence and duration that is reported to positively correlate with blood concentration of progesterone. And in clinical studies, oral and intramuscular administration of progesterone tends to increase seizure frequency. And progesterone's effect in the brain is mainly through 3 mechanisms. So activation of signaling cascades, it can regulate gene expression, and it can also modulate neurotransmitter receptors, or neurotransmitter systems, rather. And progesterone itself doesn't exert a modulatory effect on GABA-A receptor function, but its metabolite, allopregnanolone, does. So allo binds to this neurosteroid site on the GABA-A receptor itself, and it can increase the conductance of chloride that is evoked due to the binding of GABA. And GABA is an inhibitory neurotransmitter. So what we're seeing here is this dose-dependent response of the GABA A receptor-mediated current as we increase the amount of allopregnanolone that's available and present. We see an increase in the amount of, or increase in the current that is mediated by these receptors. Despite remission being such a fascinating feature of CAE, it remains rather poorly understood, and it's often treated as a secondary outcome in remission research. And existing genetic and early treatment studies tend to focus more on broad predictors and not so much on mechanistic pathways. By this, I mean we have maybe an understanding of which genes are implicated in the disease, which treatments might lead to a better seizure control, but we don't have a clear understanding of correlation or predictors as such of eventual remission. There's also a lack of a unified framework that can capture all these processes that are happening on multiple scales, and at least notably none that employ a computational tool or model to investigate this particular phenomenon. So to address this gap, we aim to answer the following question. What mechanisms underlie remission in childhood absence epilepsy? And to do so, our objectives included developing a multiscale conductance-based model of the thalamocortical network. We then integrate both genetic and structural connectivity factors into the model to investigate how these factors influence neurosteroid-mediated remission of CAE. So to give a little bit of background on what the major regions of the brain involved are, the cortex and the thalamus are key players in the pathophysiology of absence seizures. The cortex is the outer surface of the brain. It's divided into 4 major lobes. The frontal lobe is mainly responsible for performing higher order executive functions. The parietal lobe is mainly responsible for processing sensory input, and the cortex is also divided horizontally into layers, where each layer consists of both pyramidal and non-pyramidal excitatory and inhibitory type of neurons. Pyramidal neurons are generally the main source of output of information from the cortex. And the thalamus is a major relay station which modulates information between the 2 structures. And the thalamic reticular nucleus is a thin layer of inhibitory neurons that wraps around the margin of the thalamus. And the thalamus itself also contains excitatory relay neurons. So the basic idea being both structures have excitatory and inhibitory neurons of different types within them. So the thalamocortical circuit then consists of pathways connecting these 2 structures. And one of the normal functions of this circuit is the generation of sleep spindles, which are neural oscillations in mammals during non-REM sleep. And they're characterized by the 7 to 10 hertz pattern on the EEG. Now, in the disease state, this circuit has the ability to transform these spindle oscillations into the pathological SWDs. Again, the frequency of these oscillations is much lower, between the range of 2.5 to 5 hertz. So in this work, we considered various reductions of the cortical component of this circuit. In one of them, we retain just the deep layers, where we have distinct excitatory cell types in layers 5 and layer 6, as well as an inhibitory interneuron that's present in both the layers. And in another reduction, we condense all the layers into a single layer, where all the excitatory cell types are grouped into one population and all the inhibitory interneurons are grouped into another one. And across all of these variations, we use this framework to model both individual neurons as well as network of neurons. So individual neurons are simulated using a single compartment conductance-based model that uses an equivalent circuit representation of the membrane. Specifically, the membrane is modeled as a capacitor. The ion channels are modeled as resistors, and the battery represents the ionic, the driving force from the ionic concentration gradient. And the current across the membrane then has two components, one associated with the membrane capacitance and one associated with the flow of ions through the ion channels. And what we're seeing here is a standard equation that represents the change in membrane potential of a single cell. So the first term here represents a leak current. This term is a sum of all the ionic currents. The last term here represents synaptic current due to connectivity with other neurons. The kind of ion channels that you include in your single cell equation depends on the cell type, and it can give rise to different firing behaviors of the cell. And these behaviors can then define your cell, and they can range from bursting to just regular spiking, spike frequency adaptation, and so on. The synaptic current was modeled similar to how ionic currents are modeled, wherein you have a maximal conductance and you have a gating variable that is described by these equations down here. So T here is the neurotransmitter concentration. Alpha and beta are binding rate constants that represent the rise and decay of the current. So when a presynaptic spike arrives, T increases to a maximal concentration value, and then once the neurotransmitter pulse duration has elapsed, T will fall back down to 0 with the time course that's described by S of T. So we conducted this in 3 integrated parts. In the first part, we use a thalamocortical circuit reduction that represents the cortex as just a single deep layer. And this model exhibits baseline healthy spindle oscillations that transition to SWDs through this GABARG2 mutation. Now that's a mutation that impairs cortical inhibition. So it reduces inhibition in the cortex. And then specifically, we investigate the effect of allopregnanolone on SWD dynamics. In the second part, we developed a revised model of the thalamocortical circuit that incorporates cortical layers 5 as well as 6. And each of these are represented, or each of these distinct cell types represent the frontal and the parietal cortices. And then using this model, we look at how varying population sizes, strengthening connections within the frontal cortex, or our representation of the frontal cortex, how that influences allo-mediated SWD dynamics. And then lastly, we apply this, this revised model to look at allo-mediated SWD dynamics under disease conditions that are caused by sodium channel mutations. So to go a little further into the first part, we were modeling the effect of allopregnanolone on the resolution of spike wave discharges. And this was based on a work by Distesh and others from '98. It's a 400 cell network of single compartment conductance-based modeled neurons of the pyramidal and interneuron, so inhibitory interneurons in the cortex, and the thalamic reticular neurons and the thalamocortical relay neurons in the thalamus. And the network here is modeled using that framework that I described earlier. And this model achieves its disease state through a GABARG2 mutation, which reduces or impairs cortical inhibition. And we model the effect of this mutation in the network by reducing or modifying the maximal conductance of the GABA A receptor-mediated current. the effect of allopregnanolone is modeled by modifying some of the synaptic current parameters, specifically those within that synaptic gating variable. So you've got the neurotransmitter pulse duration as well as the rise and decay time constants. And these adjustments are informed by fitting our synapse model to data that's available in the literature. And this is just a comparison of what the two synapse types look like when implemented into the model. And what we're really doing is applying a relative change between control synapse activity and post-allo synapse activity when thinking about how to make these changes within our context. So for our baseline simulations, all of the GABA A synapses are of the control type. And at baseline, we see that the model displays this healthy spindle oscillations. So up here on the top, we have a raster plot that indicates, so on the y-axis, we have the cell index, and this indicates each dot represents when the neuron has spiked. We can see that the network exhibits a frequency of 8.5 hertz, which falls within that characteristic spindles range of 7 to 10 hertz. And through cortical disinhibition, the network transitions into this SWD state with the network frequency shifting to 4.5 hertz, consistent with the typical SWD range of 2.5 to 5 hertz. And of course, in the raster plot as well, we observe this synchronized bursting activity of all the populations. And then to investigate the effect of allopregnanolone on SWD dynamics, the network is initialized to the disease state. So what we're seeing here is essentially the same information in the previous slide, where the power spectrum peaks at 4.5 hertz. This is the disease state of the network. And then we apply the effect of allo to all of our GABA A synapses, and we observe this ameliorating effect where the network frequency shifts to one from the SWD range to one associated within the spindles range. And just to clarify, so the red region represents relative power within the SWD frequency range, and the green shaded region represents relative power within 7 to 10 hertz. And although I haven't shown it here, this effect is region specific and appears to be most significant in the thalamus, which in fact is consistent with how GABA functions across the thalamocortical network in the context of absence seizures. And it's interesting that we see that consistency given that we're modulating the binding process of GABA A receptor function instead of the more classical approach, if you will, of change in channel conductance. So given that not all individuals achieve remission clinically, our results seem to suggest that allopregnanolone-mediated remission may be possible in individuals who are predisposed to remission. Now that brings us naturally to the question of what factors might predispose someone and increase their likelihood of remission or not. And in fact, there's evidence that suggests treatment non-responders and patients who don't remit eventually exhibit increased frontal cortical connectivity at the time of diagnosis prior to treatment. So that suggests that there is an intrinsic predisposition, perhaps, of remission or towards non-remission. Which brings me to the second part of this work. So to consider modeling increased frontal cortical connectivity, we needed a model that can incorporate the frontal and parietal cortices. And to do so, we take our old model, specifically the thalamic component of our old model, and keep it as is. And we enhance the cortical component by incorporating cell types of the bursting kind as well as the regular spiking kind in layer 5, and we have a regular spiking population in layer 6, and then an interneuron population that's present in both the layers. And then we model the parietal cortex using the regular spiking cells in layer 5 and the frontal cortex using the bursting type in layer 5. And then to investigate how different cortical network compositions may affect allo-mediated resolution of SWDs, we consider varying the proportion of bursting and spiking cells in layer 5. Specifically, we have these 3 configurations where more bursting cells represent a greater greater frontal cortex contribution, and more, the other way, I think, right here. More regular spiking cells would represent a greater parietal cortex contribution. And then lastly, we model increased frontal cortical connectivity by modifying the synaptic strength or the maximal conductance of these highlighted synapses by a multiplicative factor. And then when fitting our new model, we assess its performance using evaluation metrics that characterize these 2 key states: healthy spindle oscillations and pathological SWDs. And once again, the disease mechanism is the GABARG2 mutation. And the model from the first part guides our quantitative and qualitative criteria for fitting. The fitting parameters that we used were the maximal conductance and the rise and decay time constants for the AMPA and GABA-A synapses for all the synapses except for those within the thalamus, given that we've left the thalamus or the thalamic component of the network untouched from the previous part. And then we employ this sequential fitting process where synapses are incrementally added at each stage. And by that, I mean, we find the best fitting GSYN, alpha, and beta parameter triplet before adding the next synapse optimized through a grid search. The baseline behavior for our model, the baseline model is configured with 75 bursting and 25 regular spiking cells in layer 5. The baseline behavior is consistent with the first part. That is, we see spindle oscillations, so at 7 Hertz, transition to spike wave discharges through this reduced cortical inhibition mechanism. And what we're seeing down here are just sample voltage traces, 1 for each population. In the case of spindle oscillations, as well as during an SWD state. And we also observe allo-mediated SWD resolution that is consistent with the previous case. So here our network was initialized to the disease state, and then this is the network activity post-application of allo, or implementation of allo synapses. So far, no changes. And again, this is at baseline frontal cortical connectivity conditions. And then we also test whether there's any difference between networks that are configured with more spiking cells versus more bursting type cells in layer 5. And we observe the same sort of behavior where there's no significant difference between the 2 network configurations. We still see this transition from spindles to SWDs. And similarly, post the application of allo, we observe allo-mediated recovery of SWDs. What this suggests is that there's no significant difference between in the ability to generate and maintain SWDs between networks that are more dominated more by bursting type neurons or by spiking type neurons, which is in fact consistent with the idea that there are different possible initiation sites of absent seizures, given that our bursting type population is representing one region of the cortex and the spiking type is representing a different region of the cortex. However, when we consider enhanced frontal cortical connectivity, allo-mediated resolution or allo-mediated recovery is only maintained in networks that have more spiking type neurons or networks with a greater parietal cortex contribution, which suggests that perhaps non-resolving CAE is influenced not only by enhanced connectivity in the frontal cortex, but also by the cellular composition of the network itself. And then lastly, looking at or considering disease mechanisms that are disease dynamics caused by mechanisms of different genetic mutations. Mutations in the SCN2A and SCN8A genes have been linked to the underlying disease pathology. It seems that these specific type of mutations can alter both channel conductance and the steady state activation and inactivation properties. Individual SCN2A and SCN8A variants are associated with distinct SWD patterns. And that suggests that different sodium channel mutations can lead to unique network dysfunction in absence epilepsy. And the SCN2A and SCN8A genes, they encode the NAV 1.2 and 1.6 channels respectively, which are critical for regulating neuronal excitability and generating the fast sodium current that is essential to action potential initiation. Now, depending on the mutation type, we modeled its effect by either modifying only the maximal conductance of the fast sodium current alone, or together with the maximal conductance of the delayed rectifier potassium current conductance. And we also incorporated these shifts in half activation voltage as well as the slope factor parameters for both activation and inactivation of the fast sodium current. And these modifications are based on relative changes between the wild type and the mutant channel activity. So in general, reducing sodium conductance will reduce neuronal excitability. Shifting its activation curve to the right would mean your channel requires a more positive voltage to open, that it makes it less active, while shifting the inactivation curve to the left would mean the channel inactivates at lower voltages, making it easier to inactivate. All in all, reducing neuronal excitability. But the opposite can also be true in increasing neuronal excitability. And then we introduced this mutation strength parameter that controls the degree of the mutation's effect. And then to evaluate how effective a mutation is in inducing a SWD state, a disease state, we consider this mutation score metric. The first term represents or captures how far your network frequency is from the SWD frequency range. And the second term captures the relative power within the spindle's range. And so with that definition, the lower a mutation score is, the more closely it represents network activity that resembles SWD activity. And on the contrary, the higher your mutation score is, the further away you are from an SWD state. And then using this framework, we investigated mutation effects on network level dynamics across mutation strengths and characterized the resulting behavior of the network using our mutation score metric. And then we implement the mutant channel into individual cells or individual populations or combination of populations. And we observed that the network exhibits an SWD state when sodium channel mutations affect mostly the interneuron population. and only at sufficiently high mutation strengths, which makes sense. I think it's similar in some sense to the previous GABART2 mutation where you're reducing inhibition in the cortex. Similarly here, the SCN2A mutations affecting interneuron populations represent a similar net effect. And although I've only shown data for the 2 of the 4 variants, similar behavior is seen with the other 2 variants. Next, we examine the effect of allopregnanolone on each of these disease states. And we once again observe this allo-mediated remission across all mutation strengths that are of the diseased state, as shown previously, and across all mutation strengths and both the variants under baseline frontocortical connectivity. Right, so increase in mutation scores would represent you're moving further away from an SWD state. However, when we introduce or increase connectivity in the frontal cortex, we push network activity further away from an SWD state across all mutation strengths in the SCN2A variant. However, for the one of the SCN8A variants, we see that happen only at some mutation strengths. And if your mutation strength is sufficiently high, increasing frontocortical connectivity has an opposite effect of preventing allo-mediated remission from occurring. So overall, our results indicate that allo-mediated remission is possible regardless of the underlying SCN mutation at baseline frontocortical connectivity. However, increased connectivity in the frontal cortex modulates remission depending on the gene, variant, and mutation intensity. Now, while these contributions provide a strong foundation, I think there are many directions for future work that can build on and extend these findings and of course, validate these findings clinically. One direction is in refining how we represent the frontal and parietal cortex. This was a rather naive approach of simply assigning a single population or cell type to represent an entire region of the cortex. One way to refine this might be to consider region-specific distributions of receptors that mediate excitation inhibition, given how important excitation inhibition balance is in pathological settings. We could investigate the effects of co-occurring mutations, which would better reflect the polygenic nature of CAE. We've considered the GABAR or the GABA mutation and the sodium channel mutations in isolation and one at a time, but it's possible that these occur simultaneously in many individuals. One could also model the effect of specific pharmacological interventions on genetic and network connectivity profiles, and specifically modeling disease-modifying effects of anti-epileptic drug treatment, including ion channel expression changes and activity-dependent circuit reorganization. I think this would be something that would extend way beyond the context of absence seizures or CAE. I think the idea of the effect of or drug-induced effects and changes, especially in reorganizing circuitry, is something that is likely to be prevalent and the case in several diseases, if not all. And with that, I'd like to thank you for your time and attention, and I'm happy to take questions. Yeah.
In one of your slides you showed you didn't see any difference between using 80% of bursting versus tonic spiking, but then when you do a different mutation, you do see a difference. Can you come back?
This one,
No, I think earlier than that.
earlier than that.
You are changing the network composition. Yeah, this one. Can you say a little bit more about why in the second mutation you see a difference in the recovery?
So in this setting, the mutation stays the same. The only thing that we're changing is how the cortex is, what proportion of bursting and spiking cells the cortical component is composed of. So on the left column of, I was gonna say row, left column of figures here, that represents a network that has 5 bursting type and 95 regular spiking cells in the cortex. And then in this case, we have 95 bursting and 5 regular spiking. And what we're seeing is, in both cases, it's possible for the baseline state to transition to a disease state when you reduce the GABA A receptor-mediated current. However, when you introduce the effect of allopregnanolone, we still see recovery from the disease state in the case where you have fewer bursting type cells. Now, it's possible that this is one thing to note is the frontal cortical connectivity increase is affecting the synapses both between bursting type cells as well as the connectivity from the bursting to the layer 6 population. I'm not sure if I've answered your question. Yeah.
I'm curious about the next slide, or I mean the one where you were modeling the degree of mutation. You had, yeah. So here, there is a very sharp transition about, at about 77.5%
Yeah.
mutation. Can you explain what happens with the dynamic? I mean, when you look at the activity of the network, and is that sort of bifurcation?
Yeah, so I think the sharpness in transition is more, at least visually, I think it comes more from how the mutation score is defined. The possible increases, or because the way the distance from the SWD range is defined is simply if you're falling within the SWD range, you have that term has a value of 0. But if the further you are away by a certain amount, we just assign a really high value of, I think I assigned 10 or something. So it could be that you see it's not a smooth sort of transition because the values are very discrete, if that makes sense.
Okay. And my other question is, there's a number of papers modeling epilepsy using neural mass models. And so I was wondering if you looked at that, or why did you choose spiking neurons instead? Because most of what you're showing is gray behaviors.
Right. No, that's a really good question. Mostly the reason for not choosing the neural mass models, I think, was to try and be able to look at because the effect of the allopregnanolone, we had information of specifics within the synaptic context, how it affects synaptic activity. I think we were able to, I think you can still use the neural mass models, but find a way of translating that into a little, I guess, less refined setting. Think about how you can map those changes onto the neural mass setting. I think it's doable for sure, and it might be. And I think even using like a mean field approach, I think you can do something similar with those type of models as well, for sure. Instead of looking at network models that are in modeling individual neuron activity, certainly. I think it just allowed a little bit of a. I think you can still argue the other way, but for me at the time, it I think allowed for a little bit of freedom on, specific cell type A types of like firing behaviors, and B, I think considering mutation effects that involve changes at the level of ion channels, I think incorporating single neurons and building a network using these was effective, and you can just directly translate that from what you see in the literature on mutant channel activity and wild type, instead of trying to take that information, find a way to map those changes and effects into a neural mass model setting. I think. Thank you for your question.